There is a bird that has watched every human civilization rise and fall. It was present at the edges of the earliest human settlements, drawn by fire and waste and the scent of things left behind. It appeared in the religious texts of cultures that never met each other. The Norse placed it on the shoulders of their highest god. The indigenous peoples of the Pacific Northwest made it the architect of the world. Celtic traditions saw it as an omen straddling the boundary between the living and the dead. From the ravens of the Tower of London to the trickster figures of North American oral tradition, no bird has accumulated more symbolic weight across more separate human cultures than this one. But the raven's hold on the human imagination may be less mysterious than it first appears. Because this bird, it turns out, has been doing something to our understanding of evolutionary biology that is every bit as strange as the mythology. Something that the scientific establishment, for a long time, did not see coming. For decades, the raven was treated as a solved problem. A single, wide-ranging species, Corvus corax, distributed across the northern hemisphere, from the boreal forests of Canada to the deserts of North Africa to the high plateaus of Tibet. Biologists noted regional variation in size and minor morphological differences, but these were treated as the expected product of a large, mobile, geographically widespread species adapting to local conditions. The raven's family tree appeared straightforward. Its place in the corvid lineage was well understood. Its internal population structure seemed to reflect nothing more than the predictable outcome of distance and geography. The story looked settled. And that, as it turns out, was the problem. Because when researchers began examining the raven's genome in earnest, not just the convenient shorthand of mitochondrial DNA, but the full sweep of its nuclear genome across populations drawn from its entire range, what emerged was not confirmation of a single coherent species with regional variation. What emerged was evidence of something far older, far more fractured, and far more consequential than any field guide had ever suggested. The raven, it appears, is not one story. It is at least two. And the boundary between those two stories runs right through the heart of North America, invisible to the naked eye, detectable only in the genome, and even then only if you know exactly what you are looking for. The conventional scientific narrative of the common raven's origins was built on biogeography and morphology. Corvus corax, the largest member of the crow family, was understood to have originated somewhere in the Old World, most likely in Europe or Central Asia, and to have colonized North America relatively recently, crossing into the continent via the Bering Land Bridge during one of the Pleistocene's glacial intervals, when lowered sea levels connected Siberia to Alaska across what is now the Bering Strait. From that entry point, the story went, ravens spread south and east across the continent, diversifying slightly as they occupied the different habitat zones of the western deserts, the boreal forests, the Atlantic coast. This was a model built on analogy with other northern hemisphere species that had followed similar pathways: a Eurasia first origin, a Beringian crossing, a secondary radiation through the Americas. It was reasonable. It was consistent with the general biogeographic template for cold-adapted species. And for a long time, the available genetic data seemed to support it well enough that no one felt the need to look harder. But certain observations did not sit comfortably within this model. Researchers working on corvid behavior and ecology across North America had noted, for years, that raven populations in different parts of the continent did not always behave as a single intermixing unit. There were patterns in call variation, in foraging behavior, in apparent social structure, that seemed to exceed what ordinary geographic distance would produce. Bird banders and field ornithologists reported that ravens from the American Southwest interacted differently with ravens from the northern boreal forests than simple range-wide variation would predict. These observations were noted. They were not, for a long time, taken as signals of something fundamental. Something was off even before the genomics arrived. The field just had not found the right language for what it was seeing. When population geneticists began applying whole genome sequencing to raven samples collected systematically across the full North American and Eurasian range of the species, the data returned a picture that reoriented the entire question of what a raven is. Genomic analysis, sequencing not just one inherited marker but hundreds of thousands of variable sites distributed across the entire genome, revealed that North American ravens did not form a single genetically coherent population. They formed two populations with dramatically different evolutionary histories, living across overlapping geographic ranges, visually indistinguishable to the casual observer, and in some areas sharing the same sky, the same roosting sites, even the same feeding territories. Researchers named them, informally, the California clade and the Holarctic clade. The Holarctic clade was, broadly, what the old model had predicted: ravens with genetic signatures consistent with a relatively recent colonization of North America from Eurasia, distributed widely across the continent and sharing close genomic affinity with Old World populations. Expected. Unsurprising. The California clade was something else entirely. Its genome did not look like a recently arrived colonizer. It carried levels of differentiation from Eurasian ravens, and from the Holarctic North American ravens, that pointed toward a lineage that had been evolving separately for a very long time. Not thousands of years. The divergence estimates, when researchers calculated them, pointed toward millions. This was not a regional variant of a recently arrived species. This was, genomic analysis suggested, a lineage that had been in the Americas, or at least evolving in isolation from Old World ravens, since the Pliocene or early Pleistocene. That was not a minor revision to the biogeographic model. That was a fundamental challenge to the assumption that the North American raven was a relatively recent immigrant. The story did not collapse. It widened into something that neither the field guides nor the phylogenies had prepared anyone to encounter. If you find yourself drawn to the moments when a familiar animal suddenly reveals a hidden depth, when the evidence steps forward and the accepted story has to give way, subscribe. Because the genome keeps finding those moments in the most unexpected places. 3 frameworks have emerged from the ongoing analysis of raven population genomics, and each of them raises questions that the data alone cannot yet definitively answer. The first is an ancient in situ diversification model. Under this interpretation, ravens colonized North America far earlier than the conventional model assumed, possibly in multiple waves during the Pliocene or early Pleistocene, when Beringian land connections existed during periods of glacial advance. One of those early colonizing populations became isolated in the southwestern part of the continent, perhaps during glacial maxima that restructured habitat zones across North America, pushing cold-adapted species northward and fragmenting previously continuous ranges. Isolated for long enough, that southwestern population accumulated the genomic divergence that modern sequencing detects as the California clade. On this model, the California clade is not an anomaly. It is a relic, the genomic survivor of a much older American raven lineage that the standard biogeographic narrative had no room for. The second framework takes a more provocative position. It proposes that the California clade and the Holarctic clade may not simply be 2 populations of the same species, they may represent 2 species, in the full biological sense, that happen to be in the process of merging. The contact zone between the 2 clades, which runs through the interior of North America, shows evidence of hybridization. Ravens from the 2 lineages are interbreeding where their ranges overlap. But the hybrids are not evenly distributed across the contact zone. The 2 genomic signatures maintain their distinctiveness at the core of their respective ranges while blending at the margins. This is a pattern that evolutionary biologists associate not with a single, freely interbreeding species but with 2 lineages in secondary contact, lineages that diverged in isolation and have only recently come back together, still in the early stages of determining whether they are willing to merge back into one. The third framework concerns the broader Corvus family tree, and it involves something that raven researchers had not anticipated finding when they widened the comparison panel. When the full nuclear genome of ravens was compared not just to other raven populations but to the genomic sequences of closely related crow species, the signal in certain parts of the genome did not conform to the expected phylogenetic relationships. There were regions of the raven genome that showed unexpected affinity to corvid lineages that the branching tree of the Corvus family did not fully account for. The data may indicate, researchers remain cautious about this interpretation, that the history of the corvid family involves ancient hybridization events between lineages that the standard phylogeny treats as cleanly separated. If so, The raven's hidden complexity is not unique to its own genus. It may be a window onto a broader pattern of reticulate evolution across some of the most intelligent birds on Earth. The landmark evidence that crystallized these debates came from a study applying high-coverage whole-genome sequencing to raven samples spanning the entire Holarctic range of the species: North America, Europe, Asia, the Middle East, and North Africa, combined with population genomic modeling capable of distinguishing ancient divergence from recent gene flow. The breadth of geographic sampling was what made the difference. Earlier studies had worked with smaller panels and had missed the full structure of what was there. The expanded dataset made it impossible to explain the California clade as anything other than a deeply divergent lineage with a long, independent evolutionary history in the Americas. The key finding was a divergence estimate between the California clade and all other raven populations that exceeded what any version of the recent colonization model could accommodate. The California raven was not a variant. It was a survivor. Not the recently arrived immigrant the model predicted. Not a regional morph shaped by a few thousand years of local adaptation. A lineage ancient enough, isolated long enough, and genomically distinct enough to challenge the fundamental assumption that the common raven is a single species with a simple Eurasian origin. The genome was not revising the old story. It was revealing that the old story had been describing only half of something far larger. Stand at the edge of the Mojave Desert in the hour before sunrise, when the color has not yet returned to the landscape and the Joshua trees are still dark shapes against a sky the color of iron. In this silence, if you are patient, a raven will appear. It will land on a basalt outcrop and regard you with an unhurried intelligence that feels, in this light, almost geological in its depth. This bird, or its recent ancestors, has been part of this desert landscape for a very long time, longer, the genomic data suggests, than the biogeographers assumed. It is not a visitor to this land. It may be, in the deepest genetic sense, native to it in a way that the scientific literature has only recently begun to acknowledge. There is something in the raven's gaze that has always suggested it knows more than it is showing. It turns out that was not mythology. It was genomics. What the hidden structure of raven populations is teaching us reaches beyond ornithology. It is teaching us that familiar things, common things, widespread things, things we believed we had cataloged and classified and understood, can carry within them histories that our categories were never designed to capture. We built the taxonomy of the living world from morphology and geography and, later, from mitochondrial markers that we trusted too completely for too long. And again, when whole genome sequencing enters the picture, the categories bend. The single species fractures into two. The recent colonizer turns out to be an ancient resident. The simple story turns out to be a compressed record of millions of years of divergence, isolation, collapse, and reconnection. This is not a failure of science. It is science working exactly as it should, building finer tools, asking harder questions, and discovering that the world has more structure than any previous tool revealed. For ravens, that structure includes a ghost division running through the heart of North America. Two lineages, visually unified, genomically ancient in their divergence, occupying the same landscapes and performing the same behaviors and carrying the same mythological weight in the human imagination, while being, at the level of the genome, representatives of a much older and more complicated history than anyone had written into the record. The raven watching you from that basalt outcrop in the desert may belong to a lineage that has been in the Americas since before modern humans crossed the Bering Land Bridge. The raven calling from the spruce trees at the edge of the boreal forest may belong to a lineage that arrived far more recently from the Old World. From the outside, they look the same. Their voices are nearly indistinguishable. Their intelligence appears equivalent. But their genomes tell different stories. Stories of different origins, different journeys, different depths of time. And somewhere in the contact zone between their ranges, where the two lineages overlap and occasionally produce hybrid offspring, the story is still being written. The ancient and the recent are still negotiating. Two long-separated threads of the same original lineage, finding each other again after millions of years apart, trying to determine, in the only language that evolution speaks, survival, reproduction, time, whether they are still the same thing. The raven has always been a symbol of mystery. It turns out the mystery was not metaphorical. It was molecular. And it was hiding in plain sight, in the genome of an animal so familiar that science stopped asking whether it truly understood it. The map of raven origins is being redrawn. And if the pattern holds, if the genome continues to reveal, in species after familiar species, histories that our older tools were not fine enough to see, then the raven's hidden story is not an exception. It is a preview of what careful science is going to find everywhere it looks with sufficient resolution. Subscribe if you want to be there when the next familiar thing reveals its hidden depth, because in biology, as in everything else, the most important truths are often the ones hiding in the most obvious places.